our next speaker is going to be Emerie Danson. She is mentored by Dr. Gold and Dr. Hauser, and her talk is going to be on analyzing differential gene expression and bee migrators to investigate parasite-host interactions.
All right, thank you all for coming. I really appreciate it. So the group of parasites I'm going to talk to you about is called Apicomplexan. So this is a phylum of single-cell parasites. And probably the most famous one is Plasmodium, because this is one that causes malaria, which is a very fatal disease in humans. And there's another interesting group in this phylum called Cryptosporidium. This also causes a disease called cryptosporidiosis. And so what this does is it infects the intestines of vertebrates. And so usually for humans, this means for some kinds of livestock or contaminated water, that's how they get the disease. And so it's gastrointestinal, and it is actually the second leading cause of diarrhea-related infant mortality in the world. And it can be fatal to children, elderly, and immunocompromised. And so this is just an overview of this parasite's life cycle. So they get ingested, they're taken in, they go to the intestines, they hatch and feed there, and once they're done growing, they go into the reproductive stage, and then they make this oocyst and then excrete back out. And so all these different life cycle stages, it's actually very hard to replicate this in the lab setting. Just for whatever reason, they tend to die off before they go through the full life cycle. And so because of this, usually for this disease, other drugs are used, usually ones that are meant to treat other parasites. But this is a problem because Cryptosporidium is not as closely related to these other parasites. And so these current drugs we have just aren't as effective in treating this disease. And so one solution we have is to find a model organism that is more related to them. And so one group that we have found is the Gregarians. And so you can see they actually are very closely related to Cryptosporidium. And so they're pretty similar. They both have an oral fecal transmission route, and they both have an epicellular life cycle stage. So they are partially invading the cells, they don't get fully engulfed. And so we would like to use this as a model. And something else that is Gregarians are just very interesting because they infect invertebrates, which is the majority of life on Earth. And so as a result, Gregarians are just this huge, abundant, very diverse group of parasites, and there could be potentially millions of ones that are undiscovered. And they also make it very host-specific, where they just sort of adapt very well to just one host that they're skilled in infecting. But scientists usually focus on studying parasites that infect vertebrates, not the ones that infect invertebrates. And so that means that this group is very understudied. And so we'd like to just learn more about them as a whole. And so the particular Gregarian that we are studying is called Bladdercola migrator. And so these infect Madagascar hissing cockroaches. And so these actually come naturally with cockroaches when they get them ordered from the lab. So we don't worry about introducing them or anything like that. They just are present. And they are harmless to humans, so that's not a concern at all. And something interesting is that these are still a single cell, but these parasites are actually visible to the naked eye. So they're just a lot easier to work with in the lab setting. So that's why they're such a good model organism. And so just to walk you through the life cycle, they get ingested as sporozoite, they make their way into the intestine, they feed, they grow, they hatch, they go into a reproductive phase, and then they create these immunocysts that are then excreted back out and looking to start again. And so there are three stages that we really want to focus on. The first is the attached stage, which is where they're directly attached to intestinal cells. And then the second stage that we want to look at is called the detached stage, where they're just detached away from the intestine. And then the last stage we're looking at is when they're in scissors in their reproductive form. And we're focusing on these mainly because they're just the easiest ones to access in the lab setting. And so we want to learn more about those different stages and what kind of makes them unique. And so the way we can do this is by studying the differential gene expression. You see, each cell in the body has the same set of genes in every single cell. The thing that makes them different is which genes are expressed, which ones are turned on or off, how often they're on or off. And so that's in our cells, we have the same set of DNA in each one, but it's a differential expression that turns it into a heart cell or a brain cell. And it's the same thing as parasite stages, where depending on which ones are expressed, it's how you get these different stages here. And so we can learn more about these different stages by collecting mRNA and building a transcriptome. So it's sort of like collecting DNA from genes and making a genome. This is sort of a similar thing with mRNA. And so the way this process works is we collect the samples from the different stages that the parasite is in, and then we send it off to a lab to do the sequencing. And so they break these mRNAs apart into these little fragments, and then the RNA gets converted into DNA, and then we have these adapters that get attached to the ends of each piece. And then these are sort of, lots of copies are made, and these copies are all clustered together on the plate. And then there's fluorescent dye that is corresponding to each nucleotide. And so a machine uses in it, based on the color, it can tell which base that is in the sequence, and that's how the sequence gets read. And so we got this done, we got sequences back. And so there's some different quality control steps we want to do to make sure that everything looks okay, everything's good to use. And so what I did is I used this program called Busco, and Busco has sets of conserved genes for different lineages. So we wanted to look at apicomplexes, since the migratory is an apicomplex parasite, and we would expect to see that most of these conserved genes should show up. And we can see that for the shape numbers here. And we also wanted to check and make sure we didn't have too much contamination from the host cockroach. And so we can discard running again, but looking for insected genes. And there's some here. You're not always going to have some amount of contamination present, but we just want to make sure it wasn't too much overall. And so since we're now confident in our sequences, what we need to do next is actually assemble the transcriptome. Because remember, we have all these different fragments, and we have to figure out what order they go in. And so this is a bit challenging because we don't have a reference genome to use to order them. Because remember, this is such an understudied parasite, and we just don't have that available to us. And so what we did is we actually used multiple different assembly software. Each one works a bit differently. Each one has a different algorithm. And so we used all three of these. And then we merged them together into one big final transcriptome. That way we're just getting as much coverage as we could. And so with that, we have our transcriptome, and you can compare the different life cycle stages to each other. And this is just showing you some numbers of genes that we got. So there's 288 genes where they're expressed more often in attached and detached stages versus scissoring. So those are genes that are maybe more important for those stages, less important for the other one. And then this graph over here is the same idea, except for genes that are down here, so we're not showing up as often. And so again, remember, we wanted to use this as a model for crispr. So what we can actually do is take these genes that we have found that are interesting or up or down regulated, and we can get those gene IDs from the migrator, and we can use this tool called BLAST, which helps you find similar sequences in other organisms. So we were finding genes that are similar to these that are in crispr. And so with that, we can get those gene IDs. And what's really cool is that online there's this website called CryptoDB, and it has databases of crispr genes and their known or predicted functions and pathways they're associated with. So that can just tell us a bit more about what these genes may be doing. And so this is just a way of quantifying some of the results. And I won't go over all of them, but I'll just go over this first sort of as an example. So looking at genes that are upregulated in this stage comparison, we have a lot of genes that are being visually expressed, and then we find a certain amount of matching genes for this freedom. And then even from that, turning 50, we only have so many results where we can guess what these genes may be doing. And so we do have some hints of what they're doing, but we don't have the full picture of this. And so in the short term, our next steps are to look at these gene results and function results and see what we can find that's related to metabolism or signal peptides. And the reason we're looking for signal peptide genes is because these are substances that are kind of secreted by the parasite into the host, and we think This may help with getting them attached to the host cell, can kind of be involved in how they actually affect and attach there. And something else is we actually just took more samples and we want to get these sequenced and added back into that transcriptome, just so we have more data to work with. And then in the long term, what we want to do is when we narrow down those genes a bit more and know which ones we're really interested in, we want to do some CRISPR knockouts where those genes are knocked out in the parasite, and we can see when they are missing, then what is the effect, so we can better guess what we're doing. And then one possibility is we may investigate different measurements with better characterized hosts, just because not only is bee migrator not very well studied, but also the hissing cockroaches don't have a reference gene either. And so again, that's that host contamination. It's harder to filter out when you don't have that reference to be looking at. And then finally, single cell transcriptomics may be cool to do as well, where you take these samples from just one cell at a time, so you can be very specific when you're looking at. This would be cool. It is very expensive, so that's why it's on stuff. And so I want to thank Dr. Hauser for all of his help in the bioinformatics analysis process, and he's been mentoring me for a long time during my time here. And then Dr. Will, of course, with the parasite lab, helping me with that side of things. And then, so Mia has actually taken the samples from the parasites, and she helped a bit with the expression analysis, and also with things right, because she's also in the bioinformatics, and that's been helping with this as well. Thank you for watching.
Great presentation. So we have some time for questions. For anybody?
Yes.
So you mentioned the sequencing of the mRNA. What platform do you use to sequence it?
So we send them to the NovoGene lab, and they use Illumina to sequence it.
Okay.
I have a question. I know this is maybe not your necessary area because you're only working more with the sequences themselves. But you said the cockroaches become infected with the bee migrator already. Are there other variants that also infect the cockroaches? Like, is there any way to know for sure that it's bee migrator versus a different variant that could be affecting?
I believe these are exclusive ones. They don't, I really don't have any parasites affecting them.
What's something you learned, like in this process, that you think you'll take with you for moving forward?
Well, I definitely learned a lot. I guess it may depend on if I work with parasites more in the future, because I definitely learned more about how they work. And I also learned a bit more just about how sequencing works, because I've heard about this process before a lot, but I didn't know all the differences between like which platform to use and like how it's actually read into it. So that's really something to learn. You have another question?
Yeah, this is really pretty fascinating to me. It's kind of a gross question, but like, how did you process the cockroaches? Did you dissect them and take their guts out?
So I don't work in the lab, so that's not my responsibility, but
Okay.
from my understanding, you do have to dissect them and cut them open to access and test them. So that's as gross as you find it to be, I guess.
So you said you just went through that one line at the table, but there was like 2,000 or whatever genes that were upregulated. But of those, you found a match. Yeah, for like 131 that have a match in cryptosporidium. Do you look at any of those in particular, any of the pathways or any of the
I can show you some examples actually if that's what you want to see. So, and this is for the gene ontology, so what the function may be. And so something interesting is that the same gene could have multiple functions it's associated with. This one I highlighted, it's involved in a few different things related to like RNA regulation.
Any other questions?
So when you get to the CRISPR part, right, you start knocking out genes, do you have a way of rank ordering your hit list So, right, I assume you're going to do something like look at the pathway that is of interest and then back out to the potential genes and see if you can figure if they're there. I mean,
Yeah, I'm not sure what our exact criteria for that is going to be. We definitely have a lot in there and know what to do. I guess it depends on what we want to approach first.
All right, let's give Emerie again. Dr. Gold, opportunity to say a few words specifically.
So it's been a real pleasure and actually honored to take, well, to Dr. Hauser and I, which are joint ventures. Dr. Hauser and I, so all the, I would like informatics stuff as Dr. Hauser's mentorship and not mine. But it's been just really awesome actually to work with Emerie this last year since Dr. Hauser's been a big retirement. She's extremely diligent and also just holds herself up to a really high standard of what she's producing. You know, she just won't compromise, and that's a good thing on what she and what her outputs, so whether it's an abstract, a data, she always wants to take it to whatever the maximum it can be. When she sends me an abstract or a poster or a talk or whatever, the slides, it's always like in a really high quality right off the bat. So it's a lot easier just to make something that's already really good, give my less than 2 cents to improve it. She won a poster award, a presentation award at the American Society of Microbiology Texas Parents Meeting recently. And I think it's really starting to pay off. And it's just been a really pleasure. And, you know, there's no book in this stuff, right? I mean, she's had to pick up all this stuff. I always tell my research students or students that work with me that the thing I like to say the most about you is that you've taken the project and made it your own. You've really taken ownership of the project. And I can tell you that Emerie has absolutely done that. And it's very clear to me. I think she's got a really bright future in bioinformatics and culture. So, the coordinator, how does that work? Not that I've done it already many times, but, you know, I was forget. Congratulations. You earned it.